Welcome to the fifth event of our Translational Neuroimaging Education Program. In this program, we aim to progress the translational application of computational neuroimaging techniques to enhance human brain health. This education program is nested within the lifespan working group of the Enigma Consortium, and this is a very inclusive program that we welcome researchers from all levels of neuroimaging skills and from diverse training background. The directors of these programs are Dr. Shalala Haas and myself, and we do have an international advisory committee. Please consider to join us our mailing list to receive notification about new updates about the of the program. We also have a YouTube channel, and there you can find our video recordings from the past events if you missed them. In addition to the YouTube videos, we also have a Twitter account. Please consider to follow us there. If you wish to share your work through this platform, please get in touch with us, and we would be very delighted to host you as a guest. Today, I'm very delighted to introduce Dr. Shalala Haas and our PhD student, Yutong Yu, from the Icahn School of Medicine at Mount Sinai and University of British Columbia. They will be sharing their recent work on brain age estimation using brain morphometric data at different scales. Their presentation will also include a demo on how to apply the pre-trained models to your own data sets. Before I pass the microphone to our speakers, I would like to mention that we have an event scheduled at October 2nd. And in this event, we invite the new PI, Dr. Junhao Wen from USC, to introduce their work on using multimodal biomedical data and genetic data to investigate the association and relationships between aging and disease. So please register for it with this QR code. Or you can go to our website directly to find the information. Now, Yutong and Shalala, the floor is yours.
Yutong, I guess you will start, and then I'll kind of jump in, and then at the end we'll go through the demo.
Okay, sounds good. Can you see my slides right now? Okay, awesome. Hello everyone. Welcome to today's talk. And my name is Yutong Yu, or Jordan, from the University of British Columbia. And I'm currently a second-year PhD candidate in bioinformatics, supervised by Dr. Sophia Frangu. And today, I would like to share my most recent research about brain age prediction with brain morphometry. So as today's talk focuses on age, and let's begin by its definition. So according to Statistics Canada, age refers to the age of a person at the last birthday. And this is the most conventional definition of our age. And now we refer this age as the chronological age. But in the field of scientific research, the concept of age extends far beyond the chronological age, so which probably includes biological age. And what is biological age? So biological age refers to how old your body parts are based on physiological evidence. And compared to chronological age, more and more clinicians and researchers realize that biological age is the better indicator of, for example, residual lifespan, functional capacity, and risk of age, and disease-related changes, and so on and so forth. So here we have an example of two people, the orange one and the blue one, and they were born at the same time with the same chronological age. However, because some of the different variations from internal or external, such as their genetic, epigenetic, environmental factors, and or different lifestyle, they actually progress through different trajectories that characterize biological development and aging at the different rates or speed. And we can actually see from this example that The blue guy developed slower, but aged faster than the orange guy. And this is some very likely that the blue one is very likely associated with some earlier onset of some severe diseases. So that's why they're very different. So now let's understand what's the brain age really means. So essentially, brain age refers to the biological age of our brains, which might differ from our chronological age. And the difference between our brain's biological age and our chronological age is what we call the brain age gap estimate, or brain age with the capital letters of AGE for short. And this concept is very crucial in studying how our brains age and how this aging is linked to various neurological and psychiatric conditions. And as the field grows, the need to develop accurate and robust models to predict the brain age becomes increasingly important. And there are also several methods to predict the brain age, including using different types of data, for example, the genetic data, cellular data, and the neuroimaging data. So for our study, we're focusing on structural MRI, which is one of the most common types of neuroimaging data to assess the brain age. And why brain age is important? Because people notice that there's an apparent accelerated brain aging, which is common in several brain disorders. And according to the 2019 report, the top two disorders considered that have most significant brain age acceleration are dementia and multiple sclerosis. And this finding was replicated in the recent report published in Nature Medicine. And in this new report, they also considered other biological age models, like models of a cardiac system, immune system, et cetera, But in short, all these findings, these figures gives us the hint that it's very necessary to look at the biological age rather than purely focusing on people's chronological age. And therefore, having accurate brain age predictive models is getting increasingly important. And our group started by focusing on youth from age 5 to 22 years. It's because this age is in the life stage that has the most dramatic changes in the brain. And one of the biggest findings from this study conducted by our colleague Amir is that we identified the optimal algorithm for predicting brain age after systematically reviewing and comparing 22 algorithms. And we conclude that the best algorithm to predict the brain age is support vector regression with a radial basis function kernel. So we use the Schaefer 400 parcellation morphometrical features to create the model and predict the brain age for youth group. And based on this finding, the optimal algorithm, we extended Amir's youth brain age models to an age range that covers most of the human lifespan from 5 to 90 years. And this paper is now published in Human Brain Mapping, and this study is the one we're going to focus on today. And the paper QR code is right here. So if you want to learn more information, please take a screenshot and scan the QR code for more information. So in this study, we collated data from an Enigma Lifespan Working Group and other public data sets. And in total, we have over 35,000 scans from healthy people aged from 5 to 90 years as model development sample. And we also collated 2 more independent samples for testing purpose. And the first one is a cross-sectional sample of over 2,000 healthy people aged from 8 to 80 years. And the second testing sample is a longitudinal sample of 377 healthy people aged from 90 to 25 years. So the longitudinal sample has 2 scans with an average interval between the 2 scans of about 2 years. And the feature used in this model include cortical thickness and the surface area of brain regions from the DK atlas of 68 parcels, and also 14 subcortical volumes from ASEC atlas. So in total, there are going to be 150 features used in this model. And with such large and diverse data set, it might seem that more data automatically translates to a better model. But is that really the case? Are there any other factors that could influence the performance of our models? So to answer these questions, we've set 3 key objectives for our ongoing research. And the first, to assess the effects of sample age range and site harmonization on the performance of machine learning models for brain age prediction. And the second is to establish a minimum sample size requirement for a stable brain age model. And last, to test the longitudinal consistency of pre-trained brain age models. So after figuring out these objectives, we're able to deliver the optimal, the best performing algorithms and models for public to use to predict the brain age. And one of our initial areas of exploration was the impact of different site harmonization strategies on the model performance. But before we look at these findings, let's just clarify what is site harmonization is and why it's necessary. So imagine we're collecting data from so many different locations and each using different scanners, like Philips scanner, for example, from University of British Columbia, and a Siemens scanner from the University of Toronto. And these varying scanners and locations more or less introduce inconsistencies into our data. So site harmonization here addresses this by standardizing the imaging data across different sites and scanners. And this process involves applying correction techniques to reduce these variations in image quality, acquisition protocols, and scanner characteristics. And the aim is to minimize confounding factors from site-specific differences also to make the data more comparable and the analysis more reliable. And in our study, we compared 7 site harmonization strategies. So the first strategy is no site harmonization, followed by generalized linear model with site as the covariate. And next is the combat GAM, COV-BAT-age with the age preserved as one of its covariates, and COV-BAT, and a subsampling maximum mean distance-based distribution shift correction algorithm. And the last one is a deep learning method called invariant conditional variational autoencoder. And in order to evaluate these model performance, we use 2 metrics to evaluate. The first metric we used is mean absolute error, or MAE. And the second metric is correlation between the chronological age and the predicted brain age. And our goal was to identify a strategy that yields a high correlation along with a low MAE. And from the figure, as we can see, very surprisingly, that the results actually showed that the strategy with no site harmonization actually performed the best, and because it achieved the lowest MAE and the highest correlation across different age intervals. So the conclusion is that no site harmonization strategy has the best model performance. Therefore, in all of our models, we do not do site harmonization. And the next, we turn our focus to understand how age and sample composition affect the model performance. So we segmented our model into different age parcels, like we create a model for every 10 years, 20 years, 30 years, 40 years, or the entire lifespan. And we just observed that as we increase the span of each age interval, the correlation across these interval generally increased. However, the MAE did not follow the similar pattern. And the tightest interval, like the 10-year range, showed the lowest MAE, while the 40 years intervals were the next in line. And consider we have to get a balance between the high correlation and the low MAE for optimal performance. We conclude that dividing our sample into two broad 40-year intervals, specifically from 5 to 40 and 40 to 90, will provide the best model performance. And this approach just provides the right balance to ensure that the model is both precise and reliable. And then we explored the necessary sample size required to achieve a stable BrainAGE model. Because despite our initial large data set, the discovery sample, which includes over 35,000 individuals, we question whether we really need such a vast number of participants to get a stable model. And to explore this, the discovery sample was randomly partitioned into 30 subsets, ranging from 200 to 6,000, with an increment of 200 each time without replacement. And we assess the model performance using this strategy. And the data actually demonstrated that the both MAE and the correlation metrics did not stabilize until the sample size exceeded approximately 1,600 individuals. So from this analysis, we conclude that a minimum of 1,600 subjects is crucial for developing a stable and accurate BrainAGE model. And this finding can provide a guideline for future research in terms of the scale of data collection required. So if you want to create your own BrainAGE model using the structural MRI data, you need at least 1,600 individual subjects to get a stable model. And finally, we evaluate the longitudinal consistency of our models. And this involved using the method, this just used the models on brain scans taken from the same participant at two separate time points. And the average interval between the two scans was about 2 years. The data we used is from Southwest Longitudinal Imaging Multi-Model Study and the Queensland Twin Adolescent Brain Study with 377 objects. And this analysis focused on how well the models performed over time across different age intervals. And we just discovered that the models divide into two specific age bands, like the optimal one we just discovered, 5 to 40 years and 40 to 90 years, demonstrated the highest consistency in the longitudinal data. And this is because for this partition, it achieved the smallest variations in MAE and the correlation between the two time points. So essentially, the differences between the MAE and the correlation at the first and the second scans are or minimal, which indicates a very strong stability of the brain age estimates over time. And this robustness is very crucial for ensuring our models can reliably track and predict the brain aging, even as the same individuals had changes over several years. Because our discovery sample mostly based on the cross-sectional data. And here we verify that the model can not only apply to the cross-sectional data, but also can be used on the longitudinal data. And to provide better access to our model, we've made our BrainAGE model available through an online calculator, where users can upload their own data from the CentaurBrain website. And CentaurBrain is a platform dedicated to open science and web-based individualized neuroimaging metrics. And additionally, you can scan the QR code to go to the page to play around your own data. But I'm also going to give the live demonstration on how to use this BrainAGE online calculation after Shalila's talk. So I think that is my part, and I will hand over to Dr. Haas.
Sorry, Shala, you muted.
I'm not able to see. It says the host isn't allowing multiple.
Yeah. Now you can do it.
Okay.
Sorry about it.
Can everyone see my screen? Okay, great. Yeah, thank you so much, Yutong. You gave a lot of the groundwork for the work that I'll be presenting. Today I'd like to share our work on developing models of network-informed brain aging using, again, brain morphometric measures. And this largely builds on the work that was just presented by Yutong. With no disclosures. So just as was described in the previous talk, extensive prior research has established that the difference between neuroimaging predicted and actual chronological age, commonly referred to as the brain age gap estimate or brain age, is a robust biologically meaningful measure of brain health. However, the majority of brain age models that have been computed as a global index that captures the totality of age-related brain changes. In one recent study, actually by Kalk and colleagues, they have developed a lobe-wise model of brain aging and showed disease-specific patterns for patients at different stages of Alzheimer's, with the largest degree of advanced aging shown in the temporal lobes, as you can see in this figure here. And moreover, in a recent meta-analysis that investigated links between brain structure and matrix-inspired cognitive domains, the authors classified structural atlases into one of the 7 Yo networks, and were able to show mapping of brain structure and cognition associations to functionally defined brain networks, reveals links between specialized networks and domain-specific cognitive dysfunction. And in this specific study, they were focusing on patients with schizophrenia. So this kind of led us to investigate whether looking at kind of a network-informed brain aging using, again, brain morphometric measures would be useful and provide more fine-grained information about changes in aging patterns that we see in the brain age across various different networks. So here we utilize open access data sets in order to build the network-informed brain age models. You can see there are quite a few listed there. We divided the data sets into First, the model development sample, which included approximately 9,300 individuals aged 3 to 92 years, with 54% female. Then an independent test sample, which included the Healthy Connectome, Human Connectome Project for Development, Young Adults, and Aging. And then finally, we also, similar to what Yutong has done in his study, looked at the longitudinal consistency of the models in the QTAB and SLIM datasets that he also mentioned, which had a scan interval of approximately 2 years. And their age range is from 8 to 22, with 55% female. Before I get into the aims, I just wanted to also note that we divided the age bins of the models for the network-based BrainAGE, for each of the network-based BrainAGE models into 2 separate age bins based on Yutong's findings, with the split being at the age of 40. And we also did not include any site correction, again, based on the best models that were identified in Yutong's work. So for this specific study, our aims were to establish the optimal number of parcels. Here we're using the Schaefer parcellation, using measures of cortical thickness and surface area that are mapped onto one of the 7 Yo networks. And we looked at different parcel sizes ranging from 200 to 1,000 with steps of 200 each time. And here the next aim was to also establish a minimum sample size required for stable BrainAGE models across different networks. And then based on the first 2 tests, we then developed the model of network-informed BrainAGE in a large sample of healthy individuals, so the model development sample, and tested their performance on an independent data set, so the 3 Human Connectome Project data sets, and then the longitudinal consistency across 2 scans from the same individuals. And the networks were, yeah, I already mentioned that the networks are mapped. The networks were derived using the Schaefer parcellation and mapped onto one of the 7 Yo networks, which you see listed up in the top right. So we have the visual, somatomotor, dorsal attention, ventral attention, limbic, frontal parietal, and default mode networks. Okay. So here we show the results looking at the performance based on using different numbers of parcels for each of the different networks. So here again, similar to Yutong, we're reporting the mean absolute error for each of the different networks and each of the different parcel numbers, as well as the correlation between predicted and chronological age or actual age. And what we see here is that having the most fine-grained number of parcels, so 1,000 parcels, seems to consistently show the best performance across the different networks, whether you're lower mean absolute error, and then as well higher correlations with the 1,000 parcels. And then for establishing the minimum sample size, again, here we randomly sampled subsets of the data and reran the models for each of these different sample sizes. And what we find here is actually we need somewhere on the order of 3,000 individuals in order to have a robust model of network-informed BrainAGE, at least when looking kind of across the different networks. There is slight variation with each of the different networks, but it seems to kind of stabilize at 3,000. Okay. And the model performance for the model development sample are shown here. So here we have the results for the testing. And what we see is that model performance is consistent across networks, with the only difference being that younger individuals have a lower mean absolute error than older individuals. but when looking at kind of the correlations between predicted and chronological age, these are relatively consistent in each of the different age bins. And then following that, we applied these models to the independent test samples, which were, again, the Human Connectome Project 3 samples for a total of 2,845 individuals with an age range between 5 and 89 years. And here, again, we see when applying these models to the independent test data sets, performance is consistent with the model development sample across networks. So for the majority of networks, the mean absolute error doesn't exceed 5.2, and the correlations are above 0.78. And again, here, we split the data into the young individuals and the older individuals, split at the age of 40. Here, finally, we're also showing the results when evaluating the longitudinal consistency of our models. And this involved, again, using the models on brain scans taken from the same participants at 2 different time points with an average interval of approximately 2 years between scans. Our analysis focused on how well the models performed over time. And since all the individuals in this sample were less than 40, we used the models for those younger than 40 years of age. Here we found consistency across networks, with the greatest difference between the 2 time points being in the limbic network, with a mean absolute error difference of about 1 year between the 2 scans. There was an average difference of approximately 0.1 in the correlation values between scan 1 and scan 2. And again, the greatest difference was within the limbic network. And as Yutong mentioned also in his talk, testing this robustness is crucial for ensuring that our models can reliably track and predict brain aging, even in the same individuals that may experience some changes in the brain over the years. So here, for most of the networks, we see relatively consistent performance measures across the 2 scans. Similar to what Yutong showed already, we also have these models online and available for you to try out yourself. Yutong will show more details of what is needed in order to run these models on your own data sets in the demo that's coming up. And there, definitely keep a lookout. There will be a preprint coming out hopefully in the next couple of weeks. Yeah, so I'll hand it back to you, Tom.
Thank you so much, Shalila. And next, I would like to give a live demonstration on how to use our Centaur-Brain website to calculate the brain age. So I'm going to share my screen again.
Everyone, as Yutong is setting up, so we will have a Q&A session after the demo. So please have your questions ready, and yeah.
Okay. So can you see my screen? Okay, awesome. So here we at the homepage of Centaur-Brain website, and on the top, the menu bar, we find the brain age model. So we will be redirected to the page. We're able to calculate brain age. And you'll notice 3 different tabs underneath. The first one is to generate developmental brain age for sample. And this tab is the very first study I mentioned from our colleague Amir, is to calculate the youth age group from 5 to 22 years. And I'm not gonna demonstrate this part because we're focusing on the second and the third. So the second tab is to use the global morphometrical measures, including 150 features from cortical thickness, surface area, and subcortical volume to predict the brain age. And here we see that we need to select the sex and followed by the age range. So I would like to mention that all of our models are sex specific. So you don't have to worry about the sex issue while you just need to split the data into different sex. And here, for example, if we select the female from 5 to 40 years, the page will pops up. What we only keep the information is your email. So other than that, you don't have to worry about your data. We will not keep your data. So even if even you upload your data for the online calculation, we're not able to see what data you uploaded. And here are 5 very simple steps. The first step is to follow the standardized free surfer recon all workflow to extract the cortical thickness and surface area features based on the DK atlas, and also the subcortical volume based on ASEC atlas. And after run the data pre-processing, the next step is to populate your data into our template. So we provide a template here, and you can download just a one click, and let's see what it is. So here's the spreadsheet, and some of the columns are yellow columns, and some of them are no color. And the yellow columns are the columns you're required to fill in your data. And for the columns without the yellow, it's optional, totally depends on you. So what we need you to populate is the site information and also the age, as well as the morphometrical features. And in this template, it has 150 morphometrical features. So it's just a lot of columns after. Okay, so back to our, oops, sorry, back to the website. Once you populate your data, upload your Excel to the website. So here I just prepared a demonstration, demo data here. And after the upload, you're able to preview your data on the right panel to make sure that this is the one that you would like to calculate, BrainAGE. And we can see this is the one that we want to calculate. So just a final step, once click to compute. It takes a few seconds to finish the calculation. And once the calculation completed, it will show up here, the message computation is done, please download the result. And here we have 4 buttons to download the different result. The first one is the predicted BrainAGE, and we can take a look. So the predicted BrainAGE, this is the BrainAGE, the predicted BrainAGE. And the first column is the order of the subject. And it remains the same order of the subjects you uploaded to the website. So for example, here we uploaded 13 subjects, and it will calculate the BrainAGE for each accordingly. So this is the first result. And the second result is the BrainAGE gap estimate. So it's just the difference between the predicted BrainAGE and the actual chronological age. And likewise, the order remained the same as the subjects you uploaded. So yeah, they're just consistent with that. And the third one is called the adjusted predicted BrainAGE. This is the one we use the linear regression to further adjust the predicted BrainAGE based on our pre-trained model. So essentially, we create the we use the linear regression to regress between your uploaded data and our pre-trained model to give a better adjusted BrainAGE. And it will just look like very similar to the previous two right here. And the last one is the adjusted BrainAGE estimate gap, and it will give the same look as the previous ones. So this is essentially the demo for the global BrainAGE online calculation, and you can choose from between female and male, and choose between the two age groups. The next part is you use the Network-BrainAGE. So when we click the third tab here, we're on the page of using the Network-BrainAGE model to predict the BrainAGE. And likewise, we need to select the sex, then followed by the age range. And it's just slightly different because the previous one, the age range is from 5 to 90, and the network one is from 1 to 95. And also, what we only need to do to provide is the email information. Here, also 5 simple steps, but in the very first step is different from the previous one. So here we still follow the free surfer recon all workflow and extract morphometrical features. However, here we extract the cortical thickness and surface area based on the Schaefer 1000 parcellation scheme. And we also attach the link to the Schaefer 1000 parcellation scheme, where you're able to see what specifically they are from this page. And we also provide the script that can extract the Schaefer parcellation from your free surfer output here. So yeah, just for your ease. And the next is still to download the template, and we provide the template on our GitHub. And this time you need to download it manually here. And likewise, we need your site information as well as the age information and all of the Schaefer 1000 parcellation features. And for the subject ID, sex, scanner type, free surfer version, they're optional. And what I would like to mention here is that this template, because it uses Schaefer 1000 parcellation, it will contain 2000 features in total. So this template is way bigger than the previous template. So when you upload your data, it just take much longer time. It may take a few seconds or even a minute to finish the upload. And we also require user to not upload the data with more than 150 subjects at a time, because that's going to be a very, very huge matrix, and the calculator performance is very limited for large files. So if you have more than 150 subjects at a time, please split into separate files and upload your data separately to get the predicted BrainAGE. And here, after the upload complete, we're able to preview the data on the right panel as well. So make sure this is the one we want to calculate, and this is. Then click the compute. It will also take a few seconds to finish the calculation. Here we go. And likewise for download buttons, and let's see what they are. Here, we include a little bit more information. So we include the site, just the basic demographic information you uploaded, followed by 7 predicted network BrainAGE for each region, for each network. Like this one's for visual, this one for 1, 2, 3, 4, 5, 6, 7. So you will get 7 different BrainAGE for each network. And next is the BrainAGE gap estimate, which is also follows the same format. You will get the BrainAGE gap estimate for each network. And also for the adjusted predict BrainAGE and predict adjusted BrainAGE gap estimate. Yeah. So yeah, this is it. So I think this is the demonstration for both of the using the global measures and also the network-based BrainAGE. So I hope this is helpful. So thank you very much.
Thank you, Yutong and Shalila. We do have a couple of questions in the chat box. I saw